Buffers. What's the first thing that comes into your mind when you hear the word buffer? Most students would say YouTube, video buffering. But that's not what we are going to talk about in this video. So we will talk about buffers in the context of biochemistry. So buffers, before I define them, I will tell you why they are important. For one, we have buffers inside our body, particularly in our bloodstream. It's very important because this buffer maintains the physiological pH of our blood. And that is very important because there are diseases associated with very low pH in the blood and very low or very high pH in the blood. And if this is prolonged, it can actually be fatal. Next, buffers are also important for the structure of our biomolecules. Okay. If you see here, this is a short clip of a protein called kinesin. And these proteins are responsible for transporting small molecules inside the cell. This clip is particularly portraying a kinesin walking on a microtubule. Now, the function of these proteins is associated to their structure. So they must maintain their structure, otherwise, they won't be able to perform their function properly. Now, it's important to keep the pH constant inside the cell, and that is caused by your buffers. Otherwise, any change in the pH level can also lead to a change in the structure of these proteins and other biomolecules, and this can affect their function. So now that you are convinced on the importance of buffers, let us now formally define them. So buffers, or more technically called buffered solutions, because they are solutions that are buffered, these are solutions whose pH values change only very slightly on the addition of small amounts of an acid or a base. So from the meaning of the word buffer itself, it's a buffer. So it prevents the effect of an additional acid or an additional base to any solution. So there are two important components in a buffer. The first component is it should be able to neutralize an added acid, and the second component must be able to neutralize an added base. And most importantly, these two components must not neutralize each other. Otherwise, it doesn't make sense because if they neutralize each other, then the two components will not be available to buffer the effect of an additional acid or an additional base. Now, when we prepare buffers, again, I mentioned it has two components. So the possible recipes that we can use to prepare buffers would be number one, a weak acid and its conjugate base, or number two, a weak base and its conjugate acid. Now, for us to understand why the recipe looks like this, or why we need two components in a buffer, we should know or we should understand how a buffer works. So, for example, okay, you all know that acetic acid is a weak acid and its conjugate base is acetate. So, let's start with a buffer that has an equal amount of the weak acid, acetic acid, and its conjugate base, acetate. So the relative levels we see here, they are equal. So we have equal amounts of the two. Now consider adding a strong acid. We know that strong acids uh, have H3O plus or hydronium ions in solution. So this strong acid is represented by the hydronium ion shown here. This can be neutralized by the conjugate base acetate. So you see here the reaction between acetate and hydronium ion. So what happens is you have some amount of acetate reacting with the hydronium ion. So this is a neutralization reaction, and the product, so you produce acetic acid and water. Now, what happens to the relative amounts of the two components of our buffer? Of course, the amount of your acetate will decrease. Meanwhile, the amount of your acetic acid will increase. However, this change should not be very big such that we expect that the change in pH would be very, very small. On the other hand, if we add a base, a strong base to our buffer, so let's start again, let's go back to our starting point. A buffer containing equal amounts of the acetate and the acetic acid. So we have our hydroxide. So the strong base is represented by the hydroxide ion. This time we have the acetic acid that can neutralize the added base. So you see the reaction here, acetic acid plus the hydroxide ion. Again, this is a neutralization reaction, and the products would be the conjugate base acetate plus water. So the production of water, that is a hint that it's a neutralization reaction. Now we see the opposite effect. This time, we have the acetic acid, the amount of acetic acid decreasing because it neutralized the added base. And we see a slight increase in the amount of acetate because it was produced in the neutralization reaction. Okay, so we now see the mechanism of a buffer. So with this, you must understand why a buffer needs two components. So here is a very short exercise. So with what I have discussed so far, you should be able to identify buffers. Okay. So the answers to these items will be in a separate video. Okay, so you answer them on your own and then check out the video for the answers in this short exercise.